All right, let's jump right in. Today, we're stepping inside one of the most important, yet kind of invisible, structures in all of modern science. We're going to explore what I like to call the library of life, this huge digital world of biological databases. So let's turn that first page and get started.
I mean, really think about that for a second. Every single gene, every protein, every little evolutionary quirk, all cataloged and just waiting for us. Now, this isn't some sci-fi movie. This is the reality that biologists are building every single day. So the real question is, how do you find the right book, or heck, even the right sentence, in a library that just keeps growing and growing?
And that, right there, brings us to a fundamental challenge. You see, in the 21st century, biology isn't just about microscopes anymore. It's a data-rich science. Thanks to some absolutely incredible technology, we're generating information like genetic sequences and protein structures at a rate that is just staggering. And the result? A massive data problem that's also, a massive opportunity.
To give you a sense of the scale here, look at this number over 1,500. That was the number of public databases way back in 2014. It's safe to say that number is way, way bigger now, with dozens of new databases and updates coming online every single year. Our library isn't just big, it's literally expanding by the second. So, what's the solution to this data explosion? Well, it's the biological database. You can think of these as the bookshelves, the catalogs, and the librarians all rolled into one amazing digital tool. They're what organizes all this information, making it accessible to scientists anywhere in the world, anytime. So let's start our tour in the first major section of this library, the primary databases. This is where all the raw foundational knowledge is stored. Seriously, think of them as the original manuscripts, fresh out of the scientist's lab. And that's the crucial point right there. When a research team discovers a new gene sequence or they figure out a protein's 3D structure, they submit that raw, unedited data directly to a primary database. It is the very first stop for any new piece of biological information. And this comparison, it just perfectly illustrates the key difference we need to get our heads around. Primary databases hold that original text, the raw sequence, warts and all. Secondary databases, which we're going to talk about in a minute, are more like the study guides. They analyze, they explain, and they add all this useful context to that original text. Now, what's so cool about this is that it shows this isn't just one library. No way. It's a global network. These three major centers in the US, Europe, and Japan make up what's called the International Nucleotide Sequence Database Collaboration. They literally sync up their data every single day, which means a scientist in Tokyo has access to the exact same genetic information as a scientist in California. It's amazing. Okay, so that's the genetic code, the DNA blueprints. But what about the molecules that code actually builds? The proteins that are doing all the heavy lifting in our cells. Well, they have their own incredible story of collaboration too. And this right here is a perfect example of that teamwork in action. So early on, you had these separate protein databases like PIR-PSD in the US and SwissProt over in Switzerland. But then, in 2002, these major players decided to merge and form UniProt. They created a single, powerful, high-quality resource for all protein information. It just goes to show how much stronger science gets when everybody works together. Which brings us to the next section of our library, the secondary databases. So if primary databases are those raw manuscripts, then these are the curated encyclopedias that help us make sense of everything. And that's the key phrase, derived from. These databases add this absolutely crucial layer of analysis. They take the raw text from the primary databases and start adding notes, identifying patterns, and highlighting the really important passages. They're what turn raw data into knowledge we can actually use. Think about it. Imagine trying to find every book on a single topic in a library this size of a city, but with no search engine. It'd be impossible, right? Well, that's where tools like NCBI's OnTrace system come in. It's this incredibly powerful search engine that links all these different databases together, letting scientists connect the dots from a gene sequence all the way to the scientific papers written about it. And the level of specialization is just incredible. You've got databases like ProCite that catalog entire families of proteins, but then you have something like Flybase, which is dedicated 100% to the genetics of the fruit fly, or DBSNP, which is only focused on tiny genetic variations. Each one is like a super specialized wing of the library built by experts for experts.
Okay, so we have this massive, complex, global system of information. But so what? Why does it actually matter? What can we do with this library of life?
This is where it all comes together. This is the payoff. Every time you hear about scientists identifying the gene for a hereditary disease or researchers designing a new drug that targets one specific protein, their work almost certainly started right here, in this library. By comparing all these sequences, they can unlock the secrets to fighting disease and understanding life itself. It is, without a doubt, one of the most valuable resources humanity has.
But, of course, there's always a new challenge on the horizon. We are now smack in the middle of the era of big data biology. The sheer volume of information being generated is almost overwhelming. Our ability to create data is actually starting to outpace our ability to store, manage, and analyze it effectively. So as we finish up here, that's the question I want to leave you with. This incredible library is growing faster than we can even imagine. New tools are being built to read its books in brand new ways. The next great discovery, the cure for a disease, or a profound new understanding of life itself, it's probably sitting on one of these digital shelves right now, just waiting for the right person to find it. So what do you think we'll read in its next chapter?